What if you could actually see the moment when the cancer cells begin to outsmart therapy? Not just they became resistant, but how they rewired themselves to survive. Today, I'll show you exactly that using our new Pathway Omics Suite. We analyzed real RNA-seq and ATAC-seq data from HER2-positive breast cancer cells that either respond to Trastuzumab or have already learned to ignore it. And what we found is a story of rewired pathways, hijacked transcription factors, and hidden positive feedback loops that lock the resistant state in place. Let's dive in. Logical mystery. Why do some cells stop responding? We started from GEO dataset, which reported paired RNA-seq and ATAC-seq profiles of Trastuzumab sensitive and resistant cells. On paper, those cell lines look very similar, but deep down, inside the regulatory network, something changes. Something that grants resistant cells an ability to escape the drug. Our goal was simple: reconstruct this hidden mechanism. Pathway Omics Suite, built for mechanistic discovery. Pathway Omics Suite brings all your omics data kind of layers, in this case RNA-seq and ATAC-seq data, into one coherent picture. It focuses not on listing the genes. It focuses on telling the story behind the data. Who are the transcription factors giving the orders? Which upstream signals activate them? And what fundamentally changes in the resistant cells that allows them to survive? This is what upstream analysis is designed for. Step 1. TRANSFAC, finding the directors of the resistance program. We start with promoters and enhancers revealed by ATAC-seq and RNA-seq. The suite uses TRANSFAC library to read the regulatory code of these genes, almost like scanning the genome for signatures left by transcription factors. In resistant cells, we see persistent activity of regulators like REL-A, FOS, retinoic acid receptor, RANX3, SOX9, and even PARP1. These transcription factors act as voices of resistance, controlling the downstream gene expression profile we observe. But who is giving them the instruction? To answer that, we step into the signaling universe. Step 2. TRANSPATH, tracing the signals upstream to master regulators. Here, the suite reconstructs the signaling cascades that activate those transcription factors. By walking upstream through the TRANSPATH network, it identifies so-called master regulators, signaling proteins that sit on the top of the hierarchy. In this dataset, regulators such as GDF15, IGF2, or LRRK2 appear as major hubs feeding resistance-related TF programs. But the real twist comes next. The walking pathway concept, the hidden engine of resistance. Now imagine this: a few key signals feed into transcription factor. The transcription factor activates a gene. That gene activates a pathway, and the pathway circles right back to activate the same transcription factor again. A perfect biological echo chamber. That's a positive feedback loop, the core of walking pathway concept. And here is the intriguing part. These loops stabilize pathological states. When they form, the cell becomes locked into the new identity, in our case, the resistance phenotype. Pathway Omics Suite doesn't just detect regulators, it uncovers those self-enforcing circles, the subtle but powerful rewiring events that makes resistant cells so persistent. When you see those loops mapped out, you understand not only who is driving the resistance, but why resistance doesn't collapse by its own. It is held in place by systematic network-level reinforcement. This is the real secret behind therapy failure. And now, why does this matter? With Pathway Omics Suite, we move beyond traditional biomarker lists. We reconstruct the functional architecture of disease, the key transcription factors, the master regulators upstream of them, and the positive feedback loops that lock the whole system into resistant state. This is a mechanism-first multi-omics analysis. And it finally reveals how complex phenotypes like drug resistance actually emerge. If you want to see exactly how we perform this analysis step by step inside Pathway Omics Suite, watch the next demo video where I walk through the full workflow. If you enjoyed this explanation of resistant mechanisms, subscribe to the channel, like this video, and don't forget to tell us which disease mechanism you want to dissect next.